This conference will now be recorded.
Yeah. My name is Jessica Reeves, Conference Secretariat at Coalesce Research Group LLC, and I will be available for any of your queries or questions throughout the webinar to help you out technically and organize the webinar successfully. So now we will proceed towards the first session of our Day 2 webinar program, that is from Dr. Pramod Stephen, the Gideons International India. Hello, Dr. Pramod. Are you ready to give the presentation?
5:30, no? So,
Yeah, hello.
hello.
Yeah, are you ready to give the presentation?
Yes, but I make up my mind to give a speech on 5:30, no? But if
No,
anybody now, yeah,
no, we already provided you the updated schedule. Yours will be the first talk and it going to
okay.
start.
Yes, I am ready to give a speech.
Okay, I'm going to make you the presenter now.
Okay, please wait 2 minutes.
Okay, okay, no problem.
Video start.
Yeah, you are the presenter now, Dr. Pramod. You can share your screen and start your presentation. Your voice is not audible, Dr. Pramod. Yeah,
Right?
yes, yes. Now you can share your screen.
ओके, स्क्रीन पर दिखाओ तो हमको शेयर करो तो कार्य की स्क्रीन पर नहीं आ रहा है. Yes, you saw me.
Yeah, it's
Hello.
audible. Hello.
Yeah, but uh, my picture is coming or not?
Yeah, yeah, your video is visible. Are you giving to a presentation by video? Like you will be presenting without PPT?
No, no. I will give a speech.
Okay, okay, no problem. You can continue with your speech.
Okay. My lovely friends, can you all hear me? I am innovator Pramod Stephen, and all things happen by the grace of God. My first innovation is low-cost animal feed by immature paddy, maize, oil cake, turmeric, salt, and calcium. For this, I got Srishti Samman. My Second innovation is treatment of anemia by bile juice. Patent number 834 oblique KOL oblique 2007. It is very beneficial for COVID-19 also. But after many conflicts and apply in drug discovery had found not done anything and they not given me any reply. My third innovation is odor-free waterless toilet design file number 31303773001. My fourth innovation is head protective cover for road safety and medical use. My fifth innovation is carbon control device for AC refrigerator, factory and homes that heat and harmful gases not go in the environment. My sixth innovation is to use water to 3 times by biofloc fish farming, and after biofloc we can use water for irrigation, washing animal homes, and finally it goes to cow dung plant. My seventh innovation is I written a book, Your Health is in Your Mouth, for any type of metabolic disorder, cure naturally, published by Novelty and Company, India, Patna, India, and Lulu.com, America. And my eighth innovation is eco-friendly poultry farm. Now I am coming on subject. My subject is immunity boost system and pre and post COVID vaccine treatment and other disease also. Introduction. Today COVID-19 pandemic is deadly big problem for the world. First things I want to say that that देखो ना नहीं कह सक रहा है. First things is to say that we have keep ourselves fit and healthy with mind and body like a soldier to fight against every kind of disease, situation, problems. Then our body and mind will be ready to face every kind of situation. Second, things is our body must have every nutrition, balance, and maintain hygiene, not more clean or more dirty, because excessive cleaning decrease immunity. Third, wear clean and pressed clothes. Tie yourself as a soldier. Fourth, be cheerful in every situation and encourage others when others Fifth, I have prepared a very simple oral vaccine. It is completely harmless and increase immunity, digest all substance needed for good health, and excrete all harmful substance by urine, stool, and sweat. Correct body metabolism. I use it since 2005 and found very good result in various problems. After the COVID-19 pandemic, I am using 2 to 3 drops after 15 days and feeling strength doing normal work with all people. I never apply for approval of the substance properly because I have no money and source to apply for that. Now I am I am saying that the world has no proper medicine and vaccine for COVID-19 and people dying like anything. According to government and WHO rule, it must go for clinical trial. But I have no facility and money for that. Eh, And money for that. I request all capable organization to experiment on this vaccine and save the world. We read it in the newspaper that coronavirus takes some change from time to time according to body, environment, and weather. If it is taking any change, then my described substance work every type of coronavirus. My prepared vaccine we can keep in every type of environment without any maintain. No need to maintain a cold chain or hot chain. And it has a long life. It can be used at any age, any and sex. Type of coronavirus. First, common coronavirus 229E alpha. Second, NL63 alpha. Third, OC43 beta. Fourth, HKU1 beta. Less common coronavirus is MERS CoV beta, SARS-CoV beta, SARS-CoV-2, which caused COVID. Structure of COVID-19. It is composite protein glycan sealed by crow spike protein. Mode of action. As we know that COVID-19 affects our mouth, nose, neck, lungs, blood, heart, and kidney. It is weak our immunity through the blood. I want to share natural method to cure COVID-19 and boost immunity by use material and method. As we know that it is sealed by protein and glycan, and bile juice secrete in our duodenum by nature. Its function is to break the protein and fat, emulsify, and support digestion in small intestine. Ethyl alcohol and rectified spirit have the capacity to absorb all ingredients and protect to the waste. If we use this combination, then people will get strength and bile juice work as stem cell and rapidly run in the blood. It helps in reducing blood sugar level and fats in the blood as it boosts function of pancreas, liver, and correct metabolism. Bile juice produced in every living creature produce the liver and no harmful substance present in it. Bile juice contain deoxycholic acid that breaks the COVID-19. We can use it as pre and post treatment process. Take any healthy chopped animal, birds, and fish bile juice and boil it. If you can add sugar or salt in it, or only bile juice and mix it with 50% bile juice and 50% ethyl alcohol, rectified spirit, or 50% bile juice and 50% corn syrup, and shake well in a bottle. And keep it for 12 hours, and after 12 hours, all particles will sit at the bottom, and we can use 5 to 15 drop upper side extract for oral admission and repeated dose morning and evening. It can develop immunity. It has the best tonic in the world. Result: I found that it cure blood clotting factor, liver disease, allergy, virus, bacteria, and many blood related disease. It is my humble request to the WHO and every government and private agency to experiment on this topic and save the world by COVID. Conclusion: I used it and found very good result. I also know some herbal plants that can cure COVID-19. No, we can use the we can use the above method. It will give result in 5 minutes. Although the taste is better, it is life-saving and vaccine also. Helped by Dr. Praveen Stephen, Dr. Sam, Dr. P.P. Stephen, and others. My substance is also tested by Drug, Central Drug Research Institute, Lucknow, and not found any harmful substance in this material. So I am very thankful to all to hear my innovation for save the world. If you have any question, then you can ask.
Thank you, Dr. Naushad. It was a very informative speech. If anyone having any questions, you can ask directly. Okay, thank you once again. I think there are no questions. As per the schedule, the next talk is from Dr. Manisha Mandal, but due to some time conflict, Dr. Kathleen Heffron has requested to place her speech right now. Can I request Dr. Manisha Mandal to allow me to announce Dr. Kathleen for her speech?
Yes, you can. You may.
Yeah, thank you very much,
So,
Dr.
will I be
Manisha.
the next person? Will I
Yeah,
be
you
the
will be the
next
next
person?
person. Yeah, you
हमारा
will
हो
be the
गया.
next
Okay,
person.
thank you. Thank
Okay,
you. Thank you.
thank you very much, Doctor. Hello, Dr. Kathleen. Are you ready to give the presentation?
Yes, okay.
Okay, thank you very much. So our next presentation is from Dr. Kathleen Heffron from University of Toronto, Canada. I'm going to make you the presenter. You can share your screen and start your presentation.
Okay.
Yeah, it's done. You can click on the button screen. That is the third button on the bottom of your screen. Yeah, it's visible. Your presentation is visible to everyone. You can start your presentation.
Okay. Thank you. So I'm going to talk today about fighting COVID-19 using plants and plant technologies. So vaccines and plants, what's all of this about? Well, the general concept is to produce vaccine proteins in plants which are routinely eaten, such as tomatoes, potatoes, and bananas. Oral consumption of vaccine proteins has been demonstrated to elicit a robust mucosal immune response in humans. Why would we make plant-made vaccines? Well, the World Health Organization estimates that millions of people, the vast majority of these being children under the age of 5, die every year from preventable infectious diseases. And these conventional vaccines can be expensive and in short supply in some countries. And by making our vaccines in plants, we can circumvent the need for syringes, refrigeration, trained medical personnel. All of these are major stumbling blocks for providing vaccines to the poor in developing countries. So this may sound a bit like sci-fi to you, but there actually are companies which produce pharmaceuticals from plants. And the first one was Protalix, which a few years ago started producing glucocerebrosidase. It's an enzyme that's used for the treatment of Gaucher disease, which is a genetic disease, genetic disorder. And they grow these huge vats of carrot suspension cells that are maybe these vats here. These are bags about 8 feet tall each, and they produce these carrot cells which express this glucocerebrosidase. So in general, the protein is this treatment costs about $100,000 US a year for a patient with Gaucher disease. But this is only pennies a glass. to treat this patient using the carrot suspension cells. This is another example that you may have heard of where plant-made vaccines and pharmaceuticals came into the picture. This is the previous Ebola outbreaks. I think this is 2012, 2015 maybe. In Western Africa, there was an outbreak, and at the time there was an experimental medicine that was an antibody cocktail composed of monoclonal antibodies made from plants. So these were made using a plant virus system. Some of it was developed here at Cornell University. And these antibodies were used to treat Americans who came back from West Africa and had contracted Ebola and seemed to work. So this is another example of where plant-made vaccines have come into the spotlight. So I think that making vaccines and other pharmaceuticals in plants provides a unique opportunity to make an impact on global health. And we can either take the plant expressing the vaccine protein, like I'm showing with the corn on the top, and completely purify our pharmaceutical or vaccine into pills and send those to developing countries. Or on the bottom, I'm showing as an example, tomatoes. We can produce our vaccine protein in tomatoes and then lyophilize the tomatoes to become a powder, which we can then resuspend into a juice or a paste and send those to developing countries. Or my favorite idea is to really just help developing countries develop their own companies and technology to produce their vaccines, which are important for their own local regions and may not be so important for the global north. So a few years ago, I got involved in a project that was another coronavirus. It was the Middle Eastern Respiratory Syndrome coronavirus. I was contacted by some Egyptian scientists, and we were very interested in seeing if we could make some kind of antiviral to coronavirus. And MERS, as you may remember, is carried by camels. There's a camel culture all across the Middle East, all the way to China. So there are literally, several billion people who live in this area and who keep camels or are associated with camels. And this virus sporadically jumps from camels to humans as a high mortality rate. So I started working with a colleague of mine at the University of Toronto, Dr. Sachdev Sidhu, who had constructed a antiviral protein to MERS. And I'm just showing you at the top left here that this is somebody's backyard in Egypt when I was visiting some scientists, and you can see that just camels are everywhere. So what my colleague did is develop a ubiquitin variant, he calls it, but it's in blue in the lower left. It's a small antiviral protein that will bind strongly to the proteinase of coronavirus, of the MERS coronavirus. So we wanted to express these on plant virus-like particles, and then we would provide these virus-like particles containing this antiviral peptide in an inhaler. So surrounding family members from someone who had contracted MERS, or maybe even the person who was sick himself could take the inhaler and have this antiviral agent provided into their lungs. So we did this by making this, using this plant virus. You're seeing on the top, it's the genomic organization of the virus. We took the coprotein and expressed it as a fusion protein along with the MERS antiviral peptide in this particular plasmid, which is really just a plasmid which expresses proteins at high levels in E. coli. So this was a plasmid that was very available in different labs in developing countries, and that's why we chose it. And you can see that even in E. coli, the plant virus would self-assemble into these shorter virus-like particles. So on the left-hand side, I'm showing the wild-type virus. On the right-hand side, you can see that there's these shorter virus-like particles, but they were immunogenic nonetheless, and they could contain our antiviral peptide. Here we're just showing the antiviral peptide. You can see that it's slightly larger on a Kamasi blue and Western blot. And so our plan is to put these virus particles containing our peptide into an inhaler and help people with MERS. Now, at the same time, just while we were working on this, the current COVID-19 pandemic came along. So we switched gears and decided to see if we could make an antiviral peptide to COVID-19. And at the same time, a number of other groups were also working on plant-based technologies to fight COVID-19. So I'm just going to mention a few of them here. Medicago is one, which is a company in Canada, in Quebec actually, and they produce virus-like particles to SARS-CoV-2 in plants. They use this using a technology that produces influenza vaccines in plants. They just switched the spike protein so that it's CoV-2 that is being virus-like particles in plants. There's another company in Ontario, which uses algae to produce COVID test kits. And British American Tobacco is right now producing millions of doses of a vaccine to COVID-19 using tobacco plants. There's also a company called Cape Town Biofarms, which is in South Africa, that's producing not only test kits, but also vaccines using plants. And finally, here's a group at the University of San Diego. They use virus nanoparticles to produce test kits, which they put on these patches. So people don't even, or these are vaccines, I guess. They put on these patches so people don't even require syringes. So I went back to my colleague, Dr. Sidhu at the University of Toronto, and he certainly started working on producing COVID specific antiviral protein. He did this in the following manner. He looked at the proteinase, which is in purple here, of SARS-CoV-2, and he found that this proteinase also had a deubiquitinase function. So ubiquitin is a protein produced by cells that plays a role in labeling unwanted proteins or even viral proteins and carrying them to the proteasome where they're degraded. And some viruses, such as coronaviruses, actually express deubiquitinases to prevent their destruction by the cell. So he found that he used phage display to look specifically at a peptide that would bind very tightly to ubiquitin and outcompete it. And so he was able to do this using phage display. And then he called these ubiquitin variants. And this is just some work he did to show that these actually can reduce virus activity because they'll bind very tightly to the deubiquitinase of the virus. And the virus cannot cleave its proteins or perform the deubiquitinase activity. So the replication cycle of the virus is blocked. So he's made all three, actually. He's made a ubiquitin variant for SARS, MERS, and COVID-19. So we have all three available, and we're producing these in plants. Thank you very much for listening.
Yeah. Thank you very much, Dr. Kathleen. It was a very informative presentation. Is anyone having any questions? Okay, thank you once again. So we moving on to the next presentation. It is from Dr. Manisha Mandal from MGM Medical College, India. Hello, Dr. Manisha Mandal. Are you ready to give the presentation?
Yes, I am. So can
Yeah.
I share my
Okay.
slides now?
Yeah. Yeah. I'm going to make you the presenter. Yeah,
Okay.
it's done. Now you can click on the screen and share your presentation. Yeah, it's visible.
Are the slides visible?
Yes, it's visible, doctor.
Okay. Good evening, chairpersons and organizers of Vaccines and Immunology 2021. I am Dr. Manisha Mandal, Assistant Professor from the Department of Physiology, MJ Medical College, India. At the outset, I would like to express my gratitude for giving me an opportunity to present my paper before you entitled Molecular Docking and Dynamic Simulation of L-hyoscyamin, eupatorium, and alkaloid L27 as potential inhibitors against 3CL-Pro of SARS-CoV-2. The ongoing COVID-19 pandemic caused with SARS-CoV-2 infection is a huge global health crisis with over 157 million infections and more than 3.27 million deaths. As of 9th May 2021, according to the World Health Organization COVID-19 report, there is a lack of specific drugs or suitable vaccines against COVID-19, and drugs that are currently available have adverse toxic effects. Some phytoconstituents from medicinal plants are worthy of investigation for formulating suitable therapeutic strategies against COVID-19 because of their safety and efficacy. The homeopathic medicines over the world, mostly derivatives of plant products, are reported useful against various diseases and might be repurposed in the management of COVID-19 associated symptoms. The homeopathic medicines such as L-hyoscyamin, a belladonna alkaloid from Atropa belladonna, eupatorium from Eupatorium perfoliatum, and alkaloid L27 from Lycopodium clavatum are antivirals with anti-inflammatory, antioxidant, and antimicrobial activities. The bioinformatic tools are useful for the estimation of pharmacological efficacy, safety assessment, understanding the dynamics and energetics of protein-ligand interaction towards designing of potential drugs against SARS-CoV-2. The 3CL-Pro enzyme of SARS-CoV-2 was chosen in this study because it is an excellent druggable target for its crucial role in cleaving the coronavirus polyproteins into functional components required for viral replication and maturation. Moreover, there is no such enzyme in human having similar cleavage action as that of 3CL-Pro that might inflict cellular harmfulness upon 3CL-Pro inhibition. Therefore, the current study aimed to perform computational validation through molecular docking ADMET analysis, molecular dynamics simulation, and free energy studies of 3 bioactive phytochemicals, namely alkaloid L27 from Lycopodium clavatum, Eupatrium from Eupatrium perfoliatum, L-hyoscyamine, a belladonna alkaloid from Atropa belladonna, against SARS-CoV-2 target protein 3CL-Pro for the management of COVID-19 using repurposed adjuvant homeopathic medicines. Now the methodology. So to achieve these objectives, the following steps were adopted. Firstly, retrieval and preparation of ligands. For this, PubChem database UCSF Chimera 1.15 and automated topology builder and repository was used. And for the retrieval and preparation of protein, RCSB protein data bank UCSF Chimera 1.15 and for pocket preparation RPBS web portals were used. For drug likeliness, ADMET analysis with Swiss ADME and PKCSM web servers were used. For molecular docking, UCSF Chimera 1.15 was used, and for Ramachandran plot regarding structural assessment for the APO and haloproteins ProCheck server were used. Now, molecular dynamics simulation for this was carried out for 1000 picoseconds using GROMACS version 2021 with CHARM 36 all atom force field. The topology for 3CL-Pro was built using GROMACS version 2021 and the topology parameterization of the ligand was carried out using CHARM General force field, that is CGenFF server. The bound and unbound 3CL-Pro were solvated in a dodecahedron box of size 10 angstrom with tip 3p water model and neutralized with sodium ions. A convergence criteria of 1000 kilojoule per mole per nanometer were applied to achieve energy minimization for a minimum of 50,000 steps to remove steric clashes. This was followed by equilibration step, which was applied on position restrained heavy atoms of the ligand along with the protein using Gen-Raster module of GROMACS. Equilibration was carried under NVT and NPT ensemble for 100 picoseconds. The position restraints were then released and MD simulation was conducted for 1000 picoseconds. Next step was binding free energy calculation. For this, MMPBSA approach was used, that is molecular mechanics Poisson-Boltzmann surface area approach. This was given by the equation as shown here, where this represents the net binding free energy being equal to the summation of energy change due to electrostatic interaction, due to van der Waals interaction, free energy change due to solvation energies, and this is the entropy contribution at temperature T. The entropy calculations were not done in this study as because they may alter the binding free energy values reported for the molecules. And regarding system, Windows 10, Ubuntu 20.04 with 64-bit operating system included NVIDIA and USD graphics were used. Now the results. First is the ADMET analysis and drug likeness. This slide represents the structure of the ligand selected here in this study, that is belladonna, eupatrium and lycopodium, and the lower panel represents the bioavailability rater showing optimal range of drug likeness properties for the selected compounds which were well within the pink zone. The optimal range of drug likeness features were lipophilicity, less than 5 size molecular weight, less than 500 gram per mole, polarity in terms of TPSA being in the range of 20 to 130 angstrom square, log solubility being less than 6, flexibility in terms of the number of rotatable bonds being less than 9, and saturation fraction CsP3 between 0.25 and 1. The values shown in this table indicated that belladonna, eupatrium and lycopodium ligands obeyed Lipinski rule of 5 for drug likeliness of a compound. Now they admit properties, that is absorption, distribution, metabolism, excretion and toxicity of the selected ligands. These were well within favorable ranges. However, eupatrium saturation value was slightly lower in terms of the fraction CsP3, which is 0.21. Nevertheless, the bioavailability score for belladonna, for eupatrium, and for lycopodium in our study was 0.55, indicating their acceptability for oral administration. Thus, in silico toxicity by ADMET showed none of the ligands belladonna, eupatrium, and lycopodium possess potential hepatotoxicity or cytotoxicity except eupatrium EMS positive. Now, molecular docking. The binding energy of belladonna, eupatrium and lycopodium, which are shown in red color, yellow and magenta respectively within the pocket of 3Cl Pro, were minus 6.9, minus 6.5 and minus 6.8 kilocalorie per mole respectively. All these three ligands occupied similar binding sites within the pocket of 3Cl Pro. Now this slide represents the 3D and 2D interaction of 3Cl Pro with belladonna in its DOX complex. It shows 3 conventional hydrogen bonds, 3 hydrophobic interactions, 3 carbon-hydrogen bonds and 2 electrostatic interactions. Now this slide displays 3D and 2D interaction of 3Cl Pro with lycopodium in its DOX complex, which indicates the presence of 3 conventional hydrogen bonds, 1 hydrophobic interaction and 1 carbon-hydrogen bond. The 3Cl Pro eupatrium complex, it displayed 2 conventional hydrogen bonds, 8 hydrophobic interactions shown in magenta and pink colored here, and 4 carbon-hydrogen bonds and 1 electrostatic interaction. Overall, the hydrophobic interactions were the most prevalent, followed by hydrogen bond interaction and then electrostatic bond. A DOX complex considers the bound state, whereas desolvation plays important role, particularly with ionized or polar functional groups in molecular interaction. Thus, characterization of dynamic interaction of protein ligand complexes with MD simulation or molecular dynamic simulation are essential. Now this slide displays the entire solvated molecules or the solvated complex and their key hydrogen bond interactions in 3Cl Pro bound with belladonna, with eupatrium and with lycopodium. Now this is the molecular dynamic trajectory analysis. The slide shows the molecular dynamic trajectory analysis of temperature, pressure, density and distance between geometrical centers of important donor hydrogen bond acceptor triplets. The temperature here attained approximately 300 Kelvin based on maximal distribution of kinetic energy that remained stable during the equilibration period. The pressure with respect to a reference of 1 bar fluctuated during equilibration, evident from large root mean square deviation. However, application was continued until the pressure reached the proper density stable equal to the experimental value of approximately 1000 kg per cubic meter and expected density of tip 3P model value of 980 kg per cubic meter. So the systems considered here in this study exhibited proper equilibration with respect to the temperature, pressure and density required for MD simulation. The molecular dynamics trajectory analysis of the angle about donor hydrogen bond acceptor and angle of gyration are shown here. The angle of gyration remained relatively unchanged about the C alpha atoms being about approximately 110 degrees and about the backbone atoms being about 112 degrees. The 3Cl Pro remained stable in its folded conformation throughout simulation at 300 Kelvin, evident from invariant radius of gyration, the average being 2.19 nanometer shown in the inset. The root mean square deviation and fluctuations are shown here. The RMSD of backbone after least square fitting to backbone for the bound and unbound protein were approximately 0.12 nanometer throughout simulation. The RMSD about the backbone fitted to ligand heavy atoms were on an average 0.4 to 0.5 nanometer. The RMSF of the ligand heavy atoms remained less than 0.2 nanometer. The RMSF of distance between backbone atoms showed no variation in structure with RMSD except the crystal structure of the free protein. Now the RMSD and RMSF again. The RMSD of distances between backbone atoms were 0.09 to 0.1 nanometer and about ligand heavy atoms were 0.046, 0.056 and 0.011 nanometer for INMW, for EWBC and X7TW, that is for belladonna, eupatorium and lycopodium respectively. And the RMSF per residue about the backbone atoms diminished from initial high values ranging from 0.2 to 0.3 nanometer to an average of 0.07 to 0.08 nanometer levels. The position restraints applied to both apo and halo protein in our study did not impose any variation in the crystal and equilibrated structure for both RMSD and RMSF of backbone atoms after least square fit to backbone and ligand heavy atoms. Now the Ramachandran plot and secondary structure analysis of 3Cl Pro bound with belladonna and eupatorium are shown in figure 8 and 9 respectively. Both in all the situation, both APO and HALO 3Cl Pro exhibited good quality model evident from 90.2% residues falling in the most favored regions in the Ramachandran plot, whereby figure 10 and figure 11 represent Ramachandran plot and secondary structure analysis of 3Cl Pro bound with lycopodium and that of APO 3Cl Pro here. The distribution of secondary structures over the entire course of simulation showed a higher involvement of turns in fluctuation events. Maximum hydrogen bond occupancy was 88.24% with belladonna complex between arginine 217 and glycine 215 compared to 82.35% maximum hydrogen bond occupancy for APO 3Cl Pro between arginine 131 and aspartic acid 289. The same values were about 119% and 100% for arginine 40 and aspartic acid 187 for the donor acceptor pairs in case of lycopodium and eupatrium respectively. 17 salt bridges were formed for each of 3Cl-Pro in complex with belladonna and eupatrium, while 3Cl-Pro lycopodium and apo-3Cl-Pro generated 16 and 15 salt bridges respectively. Arginine 131 formed maximum number of salt bridges involving aspartic acid 197, aspartic acid 289, glutamic acid 290, both in presence and absence of complexation. Now this slide depicts the SASA, that is solvent accessible surface area and per residues values respectively. And figure 12C and D, this shows the number of intrapeptide and peptide water hydrogen bond respectively. Overall, the SASA remained stable throughout the process of simulation, and a large number of peptide water hydrogen bonds compared to intrapeptide hydrogen bonds were found due to the dissolution of solutes in large number of solvent molecules. Now the slides here, that is represented by figure 13, 14 and 15. They show 2D interactions and their fractions for the simulated complex of 3Cl-Pro with belladonna, with lycopodium and with eupatorium. So here it indicates that the interaction displayed different number of interactions and patterns of interactions compared to the docked complex due to the dissolution of the complexes in large number of solvated water molecules compared to the docked complexes. This slide shows the free energy of solvation, the non-bonded interaction energy and the enthalpy here. So overall, after calculation, now the coming to the conclusion, here in this study, the net binding free energy of 3Cl-Pro eupatrium was more favorable than 3Cl-Pro lycopodium and 3Cl-Pro belladonna. Van der Waals interaction mainly contributed to the net binding free energy, followed by free energy of solvation and electrostatic free energy component. The 3Cl-Pro eupatorium complex represented lowest net binding free energy of -1 to 1.36 kJ per mole, conferring eupatorium greater receptor binding potentiality. Other ligands, that is lycopodium, with net binding free energy of -114.17 kJ per mole and belladonna with net binding free energy of -78.96 kJ per mole also demonstrated favorable binding free energies with SARS-CoV-2 3Cl-Pro, although higher than that of eupatorium. So it suggests that all the ligand molecules achieved thermodynamically stable and strong complex with the SARS-CoV-2 3Cl-Pro. Moreover, the physicochemical and pharmacokinetic properties interpreted with ADMET and bioavailability score indicated their suitability as potential drug candidates against COVID-19. Thus, the current study authenticated by molecular dynamics simulation and docking analysis the inhibitory property of bioactive antiviral phytocompounds belladonna, eupatorium and alkaloid L27 from lycopodium due to their good binding affinities and energetically stable complex formation potentiality with 3Cl-Pro of SARS-CoV-2. So that was all for my presentation. Thank you. Thank you for your patience and thank you for your hearing. That was all.
Thank you, Dr. Manisha Mandal. It was a nice presentation. Is anyone having any questions? Okay, thank you once again. I think there are no questions. We will move forward for the next presentation. It is from Dr. Mohammad Nashad Khan from Jamia Hamdard, India.
Okay,
Hello,
thank you.
Dr. Narshad.
Hello.
Yeah, are you ready with your presentation?
Yeah, I'm ready. Now
Yeah,
I share.
okay. Yeah, I'm going to make you the presenter first. Yeah, it's done. Now please click on the screen and share your presentation.
Now good.
Yeah, it's visible.
Wait. Mm, Now good, huh?
Yes, you can proceed with your presentation.
One more. Hello, I'm Dr. Mohammad Nausar, Department of Biotechnology, Jamia Ahmedabad, New Delhi, India. My today presentation in virology and immunology comprehensive whole genome sequence, computational approach to identification of potential targets for antiviral and vaccine candidates. So basically, I am talking about not a COVID and not another virus. My research area only focusing on chikungunya virus. So, on behalf of my previous research, I am talking what is the chikungunya virus and how what up to date about the chikungunya virus. The chikungunya virus is a one was described in during the outbreak in southern Tanzania in 1952. It is the RNA genome, RNA virus that belong to the alpha virus genome genus of the family probability. So many person are suffering from the chikungunya virus, maybe like the bending type nature in but in fingers, fingers joint and all the joints in your body. The proximity of mosquito breeding site and human habitat is a significant risk factor. This is a major risk factor of the chikungunya disease transmitted vector is the mosquito, basically the Aedes and Aedes family of mosquito. And currently, no viral, antiviral, and vaccine are available till date for treatment of chikungunya virus. Now, what is the vector of transmission of chikungunya virus? Basically, on the earlier slide, I can discuss Aedes aegypti and Aedes albopictus are two basically responsible for the transmission of chikungunya disease. Also transmitted dengue virus. Larva developing discarded tribe and household container. Aggressive daytime biting mosquito. Aggressive daytime biting mosquito. Basically the Aedes aegypti and Aedes albopictus. The transmission route of the chikungunya virus basically the orthoponic transmission, person to mosquito and mosquito to person. So first, basically, the Aedes aegypti is of the mosquito virus transferred to bite to human being. After that, maybe disease are responsible transmission and basically joint and training and different clinical feature I can discuss in next slide. Basically, distribution of outbreak of chikungunya disease. The chikungunya virus first described in Tanzania earlier. I can describe and more than 50 years the outbreak in Africa and Asia and some American country. As previous research, here Asia and America were the regional most affected by the chikungunya disease. And since 2004, it is an emerging disease and everywhere they can transmit it
country by country in the America and Caribbean were reported 185,000 cases. The cases in Brazil account for 90% in the region of the America. Pneumonia outbreak were also reported in Sudan in 2080, Yemen 2019, and more recently in Cambodia and China. In 2060, also reported that. So, country with reported transmitted chikungunya virus, many countries are affected with chikungunya. The current map showing where is the chikungunya virus is affected. So basically, uh, India, America, Mexico, Mexico also, yes, Brazil, and and here also some part of the UK, UK region, and here South Africa, many countries, more than 50 countries are affected with this disease. The clinical symptom of the chikungunya disease, basically those who are person suffer from the chikungunya disease, and majority 70 to 90% infected people are symptomatic. Incubation period usually 3 to 7 days, then 1 to 20. Some clinical symptom may be appear, sometimes may be not appear. Primary clinical symptoms are fever and polyarthritis. Basically, those who are person are suffering, basically starting with the fever. After 2 to 3 days, then we start with the swelling and blood. Arthritis usually bilateral and symmetrical. Sometimes within week you lost the arthritis, but with joint pain and other symptoms, but many cases more than we are remaining and not lost easily. So this is a very hectic. And other common clinical signs and symptoms such as headache, nausea, arthritis, conjunctivitis, nausea, vomiting, mucopoplar also responsible for the chikungunya disease. Basically, chikungunya disease is diagnosed by different methods, and especially here described the culture per virus. And culture per virus, you can diagnose how much plague are formed in human serum. And second is the RT-PCR. RT-PCR per viral RNA, RT-PCR through the specific primer. I can discuss in next my work in RT-PCR and IgM ELISA and detail the so the data like clinical data and what is the clinical feature and how. But here is just basic introduction here. RT-PCR is within a specific primer and serology for IgM and neutralizing antibodies through the IgM ELISA. You can first check by IgM ELISA, but IgM ELISA so after 4 to 7 days positive result. But in RT-PCR early you find the concentration of virus in blood. Serology for four driving viruses quantity, antibody titer, and paired sera. Immunohistochemical study, another method of immunohistochemical viral antigen. It is a very complicated, but there are artificial IgM, ELISA, and cultural virus. Almost virus should be handled. All chikungunya virus is handling in the BSL-3 and proper safety and precaution with researcher. So next is here initial assessment and treatment. So this is chikungunya disease. I'm earlier told no specific antiviral therapy treatment is supported. Access, hydration, and hemodynamic status. Evaluate for other serious condition and treat or manage appropriate. Basically, this chikungunya virus is a treatment based on the symptomatic. Some medicine and some like fever, such as paracetamol, aspirin, maybe also used for the reduce the fever and pain. Medicines such as paracetamol and acetaminophen are recommended to pain relief and reduce fever. And given the similarity of symptoms between chikungunya and dengue, maybe very distinct and treatment of this. In area where both viruses circulate, suspected chikungunya patients should avoid using aspirin or non-steroidal anti-inflammatory drugs. Until which time a dengue diagnosis is ruled out, because in dengue this medicine can increase the risk of. So vaccination against chikungunya. There is no commercial vaccine available to protect against chikungunya disease, while there are several vaccine trials made to 2020, and some are in various stages of clinical trial. Some maybe the third stage, some in second, some maybe in fourth stage. Yeah, work and is still going on. So there are still several years away from the being licensed, enable to the public prevention of infection by avoiding mosquito bite is the best protection. So especially genome structure, genome structure, this virus is structural and non-structural protein. This is the non-structural protein, NSP1, NSP2, NSP3, NSP4, and structural protein, capsid, E3, E2, 6K, and E1. This is a small 6K small protein, and E1 is the envelope protein. But the non-structural protein, CA, NSP2, NSP3, all are responsible for the replication in our virus in host. And here is the envelope protein, basically responsible for the attachment and the transmission of your virion in the host. So this is the primer used for the detection purpose. How to chikungunya detect when the person are suffering from chikungunya? First, chikungunya this virus. First, take the blood serum, after that isolate serum, and then this primer 294 base pair already I am published in this primer one paper molecular characterization of chikungunya virus in molecular and immunology 2020. This is the primary PCR for detection, and second is the here 913 for the phylogenetic. This is basically for phylogenetic. So the detection by RT-PCR, they are 294 base pair, and those who are sample are positive. And so here the clear band after the agrogel 2% agrogel running, and here the DNA ladder and 294 base pair clearly show the band. Then those who are light band, more visible, maybe title high in compared to others. And this is the list of primer for whole genome. In this my study, all those who are positive this virus and whole genome through the primer working, and list of this primer working, list of this primer also share with you. This is 22 pair of the 22 pair of the forward reverse primer, and after that combined and single genome. Here is the implication of the whole genome, and I earlier told 21 days and 21 fragment of the primer. This is the one fragment here, 55 days here, and do all this like the 21. This is a clear band. So after that, they combine it through the tools combined in the one sequence. And my study here in North 2000, data is North 2000, India 2000 during 2050, and reported the number of 325 clinical samples enrolled in the for chikungunya patient, and male also 142, found that as is 161, estimators and 43, and headache 233, joint pain, joint swelling. So basically, purpose of this clinical feature is joint pain and headache, also increases in number when the enroll in patient in 2006 North India during 2016. And here, after the finding the molecular laboratory through the diagnostic assay, IgM ELISA and RT-PCR, and find out 92 is the positive by IgM ELISA and RT-PCR 29. Earlier, I described the primer RT-PCR primer 294 BCR and Aerogel. The total positivity is 118. And three samples were positive by both IgM serology and TB and RT-PCR sample number, and this sample number is my database, and HPC 243, and these three sample ID is both IgM and RT-PCR. Reason of this may be early detection of this. Another compared to positivity and negativity, how much positive after the rashes and how much is negative after the rashes. Those who are all found in enrolled patient found in rashes in body, but not all is the chikungunya positive. Found in the rashes 142, but 47 is the only positive by both cases. And here headache 233, but only 63 6 are found positive by RT-PCR and IgM. Now joint pain 304 and 78 and 78, but basically here purpose of showing this data only headache and joint pain, joint swelling are more English significant, and that is indicate the chikungunya susceptible patient involved in patient. And after that, three whole genome. After that, those who are positive, three sequences, three patient samples, those who are high titer of the chikungunya virus, and isolate the serum, blood serum, and then amplify the all whole genome sequence, and finding compared to the prototype. Here ECSA is the African strain prototype of this virus, and compared to the circulating strain to this. And here is the accession number and my three of accession number and in the submitting and CBI. So finding the what is the nature of changes. So basically here which position of amino acid and what amino acid is changes. And so basically here NFP1 is non-structural protein 11 percent changes, and NFP2 is the 5% changes offer, and NFP3 is the 50% changes, the NFP4 7% offer. So basically in all this NSP2 less mutation find in compared to others. And NSP also responsible for the adequate protease activity. The protease activity also helps in the replication in hot anywhere. Now another is the structural protein, and this is a structural protein. The capsid is HT3. So basically, purpose of this same like the three whole genome my accession number of this virus and submitted in the CBI, and here prototype African strain. Compare to this each and every amino acid changes. So basically E3 the less mutation has 6K to E3 6K and capsid. So basically less even also less mutation. So this is and after that finding the mutation, and then find what is the changes in nature of the protein. It's basically protein nature also responsible for the antigenicity and responsible for responsible for replication and anything. Protein is basically building block of the and this what is the change in nature after mutation? So suppose here is the my three original sequence circulating strain and here prototype. The prototype is basically no major change in hydrophobicity, isolate electric point are a little bit increase compared to the prototype here. And basically most of these are similar, not major changes are found. And fever and human are increases. And in stability index also little bit down in this, but all this maybe similar. And here the NSP2 similarly, alphabetic index are basically no major changes after the mutation. And average antigenicity basically no major changes. Some after some protein. And similarly, like the protein also, and comparison to strain and finding the what is the nature of the change in this and cascade here, look at this one data here. Hydrophobicity similarly not major changes in isolate point is no major changes in stability indexes, a little bit increases, but fluctually, not stable, but fluctually maybe so the increases aliphatic indices. Similarly, I've seen basically similar three one to create function and average intelligence. In conclusion, I can describe what is the major changes in this. So after that, these three whole genome sequence in clustering to the same virus phylogenetic analysis. This circulating strain, how circulating strain, how to belong from the other which year and this. So finding that here not clear visible, but here Uganda, South Africa, and India 2014 strain also the similar like the 2006. And finally here epitope prediction. After the mutation of the gene, protein, nucleotide, what is the maybe epitope variation or not? So conclusion here, NSP2, NSP, here is the epitope number, this and this, NSP3, and here is the prototype. So conclusion this is NSP1 and NSP2 epitope variation. Small 1 and 2 variation and position are also variation compared to prototype. And also here antigen each gene has a little good variation. Here 6K no major changes. E2 and E3 also. So E3 little bit one similar form, 1 and 2 not major changes. And conclusion of this 327 nuclear sample suspected case enroll and find out total positivity 118 among these immunization 92 and RT-PCR 29. That is in joint pain are clinically significant. That is those who are persons suffering from the pneumonia virus and that is in joint are basically fine. Maximum amino acid variation in NF3 and E2 protein, not to other protein are very in compared to 2016 and prototype. Not the other phylogenetic relation close with 2014 and India, Africa, Uganda, Chile, and antigen variation in very small E3 gene, not a major NF1, NF2, and EC variation also found. So expected outcome in this study to find that those who are structural protein are not major variation, then target for drug discovery and structural drug discovery, and if the protein are stable, then targeted the ligand based pharmacophore and the molecular dynamic simulation and molecular docking to find out the suitable inhibitor. After that, then in through an in vivo stabilize this screening molecule are suitable for this antiviral property against virus. There is a vaccine candidate, and then E1, E2 are involved protein and so E1, E2 no major variation. So in behalf of the umbrella protein, you can design peptide-based vaccine like the MSC calculation, MSC1, MSC2, and help in what is the changes in the given the this dose and what is the nature of the this plasma and induced in the mice. So my overall study in this and end up with my presentation. And remaining any question about me regarding this anything, you can contact me or talk by email ID and my WhatsApp number also here. Thank you.
Thank you, Dr. Naushad Khan. It was a good presentation. Is anyone having any questions? Okay, thank you once again. So moving on to the next presentation, it is from Dr. Muhammad Ibrahim Said, but he requested us to postpone his speech to the last one. So I will request to present Dr. Asit Kumar Chakrabarti to present his speech. Hello, Dr. Asit. Are you ready with the presentation? Can I request you to present your presentation now? Hello, Dr. Asit. Hello, Dr. Asit. Am I audible to you? Your voice is not audible to us.
Hello?
Yeah, hello, Dr. Asit.
Yeah, there is some technical problem.
Okay. Can you present now?
Yeah, you just go to the presenter or
Yeah,
the
I'm going
share.
to make you the presentation. I'm going to make you the presenter. Just a minute.
Yeah.
Yeah. Yeah, it's done, doctor. Now you can click on the screen button and share your presentation. Yeah, it's visible. Perfect.
Have you seen my slide?
Yeah, it's visible, doctor. You can start your presentation.
Okay. Hi. I thank the Vaccine and Immunology Conference webinar organizer for giving me chance to talk. And I am Dr. Rosit and I retired. And so I am now visiting professor in a college. And I just did some bioinformatics work on coronavirus, and you all the world now know that India has a very devastating condition, and we are losing almost or sometimes higher than 4,000 people
with India, and not only that, so many people in the hospital, there is a shortage of bed, shortage of oxygen, shortage of even wood to burn the dead bodies. And they now throwing into the Ganga river dead bodies. And hundreds of dead bodies are floating in the Ganga river. They show the picture in the TV today, and I am very much shocked. I have no mood to give a talk in the conference because I think my talk is have no value in that situation. And India needs worldwide help to get rid of from this pandemic. And whatever, I give you a silent picture of coronavirus so that it may be helpful to some, and particularly the vaccine problem. So coronavirus structure, we know that this is the spike protein, and this spike protein was targeted for vaccine development because this protein binds to our lung cell, that AC2 receptor, and we know all those things. And here is the RNA and some RNA dependent RNA polymerase and other few proteins, small protein A and E, and those are structural proteins, spike, and apart from that, it processes two big proteins in same reading frame, and they proteotically cleavage into 12 polypeptides. So those polypeptides are actually important because they have a functional role. So I'm coming to that, and but here some precautionary slide. You must wear a mask, your control sneeze and wash your hands, and also lockdown is important. So, because this is the very pathogenic virus. And here I will show you the structure. This is the open reading frame, A and AB. Altogether, it is about 7,096 amino acid peptide unless there is a deletion. And this is the S protein. It is the structural protein that is important. And most vaccine made with this protein. This is about 1273 amino acids, and sometimes deleted 1 to 3 amino acids. So in that case, 1270 amino acids. And here is other small structural protein E and M, and other some protein function not very much known, but they have some role, and sometimes their mutation is predominant in case of disease. So coronavirus is three stage, very much acute stage where maybe death is inevitable, where actual 3% and where you need very strong medicine. And other is very much mild, and you essentially don't need much drug except some cough drugs for cough or vitamins and this and that. And others, even you don't know, and asymptomatic, non-asymptomatic, you don't know that you are infected because you have. So why those kinds of things due to the variation of our immunogenic power of the human. So whatever that, we all targeted S protein for vaccine development, and we want to show you today that these S proteins mutated habitually, and in that set about 3 deletion and 6 mutation, so that the vaccine candidate, which is made in vector, the gene is cloned in vector, adenovirus vector, and then that vector was injected into our body so that the DNA converted into messenger RNA by our RNA polymerase, and then they translated into protein, and that protein then proteolytically cleaved and immunologically makes cells to produce some immunoglobulin so that that immunoglobulin bind to virus, and that way we protected. Now, if there is a too many mutation, then those immunogenic fragments, they produce the antibody. They will be different. And in that way, we see today that our people who gave the vaccine, those vaccinated people are infecting with the virus that is the mutated virus. So it is very much sad thing. So we showing that Maharashtra, this red picture is very much affected, and then we show this is the life cycle where it is enter into the coronavirus ACT receptor. Then this is the positive sense RNA. They produce, give the translation to some protein directly, and then those proteins degrade the larger proteins into the RNA-dependent RNA polymerase, RNA topoisomerase, RNA helicase, and those have different functions. And we know that here the drugs Lopinavir, they are targeted with 3CL Pro. We know there is some lady talking to 3CL Pro, some drug, a plant drug, but that is very important because this drug is now used. And here we see RNA-dependent RNA polymerase, that is NSP12. This is inhibited by well-talked remdesivir. We are in India lacking the remdesivir. It is very acute situation in India. We need that whole world should supply us remdesivir. That is the message. It is we are losing 4,000 people every day. And now these positive cells RNA also they produce negative cells RNA full length, and they then again produce the smaller subgeneric RNA, and those are then packaged and make the proteins and then package into full virion. They infect, then they escape and infect more. So we have different steps where we can target the virus, but today I will talk only the S protein, surface protein. But I will talk some few points that NSP2, the proteolytic product, that is actually RNA topoisomerase, and we know that topoisomerase is a well-targeted drug in bacterial infection. So, I show you here that NSP16, which has similarity to a rRNA methyl transferase and 2-O. So that is one of the our finding, and also that NSP2 has similarity to RNA topoisomerase, not RNA topoisomerase, but DNA topoisomerase species, and also type 1 from a different bacteria, brucei, some parasites. So that way we say that is the similarity, and it's projected. This is NSP2 is a RNA topoisomerase in a different domain as ATPase domain, nicking domain, ligase domain, topoisomerase, RNA domain, and so that could be targeted, but we need some assay, in vitro assay. I told you that I have no job, permanent job now, so I cannot do it, and I have no grant, so someone to do. That's why I am showing. So we did some of this work, and other important function is NS230, which is RNA helicase. We are cumulative start. We are talking about this is a capping methyl transferase with methylate guanine. And not only that, this enzyme has similarity to L6 and L9 and the ribosomal protein. So our hypothesis is these proteins like NSP30, NSP2, and NSP16 may be both to ribosome, and they some way do meet the RNA genome methylation, and also they may change the mitochondrial RNA methylation and inhibit the protein synthesis. And this is one of our hypotheses as another mechanism of problem, ATP synthesis inhibition and septic shock and death in corona infection. And so it is not true. So in summary, we are talking about these three things, NSP16, NSP13, and NSP2, that we did some work we published. But I am talking today the vaccine, because this is a vaccine strategy. I told you that S protein was engineered in Covishield vaccine, where is the adenovirus vector. So that is the DNA gene was inserted into vector, and was that vaccine was injected into our muscle, and they change into messenger RNA, into protein, proteolytically cleaved, presented in the macrophage, T cell, B cell, and that way we got the VDJ recombination in our genome, and that way specific vaccine immunoglobulin is, IgG is produced that bind to the virus or the viral protein, and keep studying it, and that way we protect it. But if that protein is mutated, then how it will work? So I am showing you that is the S protein. This is the receptor binding domain which is bind to the ACE2 receptor. So we find here some of the data from other source. We see lot of mutation is here, and that is obvious because RNA virus like COVID-2 COVID-19, they mutated 1-2 per month, 1-2 mutation per month, and after all those mutations, only 20% change amino acid changes happen, and from this total amino acid changes, another 20% is raw, different amino acid changes that change the protein structure. So we see here that D614G. So everybody now know this 614G, 614 position D aspartic acid changing to glycine. We all know this mutation in coronavirus spike protein because that mutation changed 70% infectivity, and other mutation in the ribosome binding protein recently discovered, that is N501Y, N501Y in the London or UK variant. And now in India, all are claiming that another mutation, 484 mutation, and there is and also 452 mutation, that is double mutant, another double mutant that is claiming that virus immunoescape mutant, and also there is some 20% transcription increase. So, in my opinion, whatever the things going on, this B648G mutant now everywhere. You cannot find that normal anymore. So, an N501Y mutation is about 25%. And the other Indian double mutant, maybe you've seen in India, but I have not checked those data. Only single mutation I checked, no double mutation. And I have checked because I have no access to those sequence data. But whatever I will show you, that is very astonishing. Other important thing, the furin cleavage, the S protein at the S1, S2 boundary, their cleavage is occurred where RRAR sequence is there here, RRAR. This is the other COVID, and it is the COV2. So COVID-19. So this important position is essential. So I will show you that S1, S2, as well as the receptor binding domain, there also some mutation is appearing other than Indian double mutant, and mostly in the USA, because I got the most US data. Here we see that different clade will show you that here is some different virus, and here is the human virus. But you see here in the initially you won't get the much clade value, but they already divided according to clade A, B, C, D, A, B, C, D in different countries and even in India. But those mutations, so many mutations that and their penetration is so much changes that now new rule must be there. Sometimes that people classified as 19A, 19B and 20A, 20B, but I have not seen that this 21A and 21B, but that must be some good laboratory do that, because it is total chaos in the system of Israel Claude 26 is maximum in Israel, as you saw here, but you see there is some enhancement of the population, but then it is reduced. So here monthly sum is going up, and that is the point that the accurate value is changing. That means mutation is constantly going on. Now you have to do sequence, and you have to follow the which is the most virus that is pathogenic and immunoscape and devastating, means untreatable. So that you have to do it. So here I show you that plot at A, plot B, where some mutation is the RNA-dependent RNA polymerase, that is PL famous mutation. This mutation is coincide with the death rate. Means if that mutation is there, death rate is high. Similarly, in the S protein, we see clade A, clade B, BG mutation as I talked, and then N protein, nucleotoxic protein that binds to RNA. There is also a mutation that is under some plot value. I will come in later. And open reading frame 3A, possibly some transcriptional activator. There are also some mutation that associated with the plot A and plot B. This is the primary. It is the March 2020. Now, present story is totally complicated. Although complicated, this DG mutation and PL mutation must be there. And those are the death signal. Those are the death signal that I wanted to tell you. Now, if there is a mutation, if you take the COVID vaccine and you see in that COVID has the S gene there, you see dose 1, 0.04% is reinfected, in the dose 2, 0.006% reinfected. So there is a reinfection, but it is impossible for reinfection without mutation. That's my point. Because it is the immunology, it is the math, and our body cannot do any wrong thing. So that means mutation rates is going on very highly, and mutant is stabilizing in the system. So drug may be changed, vaccine may be changed, and I show you some of the data. Here you see the recent some data from USA and some other part, but mostly USA. You see DG mutation. Only one in Egypt there is, I got the D, aspartic acid, and that is the control China strain D, and all are mutated. So everywhere is the D614G 514 mutation, and that is highly infectious virus. There is a no change. And now the England, the variant, you see among them also Y mutation. These are the same clone, same sequencing. So you see here, and here you see 484 EK mutation. The K is changing to Q, that is Indian variant. And there is other mutation also, that is the double mutant. But you see there is very few. I take only few examples, but you can exactly predict what is going on in the scenario of virus mutant propagation and disease increase process. And you see the deletion, immunoscape mutant. You see the same virus also has deletion mutation too. Amino acid 69 and 70. And here also mutation, another amino acid. They give just cross because they don't know what is going on. But anyway, the 869 deletion, histidine amino acid and valine 70 deletion, they must be there if it is immunoscape mutant. And also there N501 mutation and D640G mutation, and mostly in the USA. So those are the papers that published. I am not talking now. What I see, the Y145 deletion has not been talked about. That's also deletion. That is in the 145 position. So they are stabilizing here. You see clearly. So must be there is another mutation here. You see there is also Y, and that is in the Italy. And I don't know what is going on in Italy. And still in the USA, another mutation D, alanine versus aspartic acid 570. Same clone, same sequencing I am showing you. And another two, you see P688 and P716I. You see there is predominating in the USA. So we don't know what is going on actually. And 981, another mutation in the same clone. And you see B1188, another mutation. Those are the normal. But those have the G mutation. And but what is I am talking about now, you are clearly understandable by seeing this picture. These so many deletion and mutation here. 1, 2, 3, 4, 5, 6, 7, 8, and 3 deletion. 1, 2, 3 amino acid deletion. So how this virus can be protected by same without deletion and mutation antigen and vaccine? It is impossible. So we will find that those vaccine, Coalesce vaccine, and also Sputnik vaccine, Russian vaccine, will be no way good only if there is vaccine is detected in between these two. Because vaccine peptide is about 30 amino acids. So if the immunogenic amino acid like the valine, leucine, isoleucine, and phenylalanine, they are in between this mutation and they selected the vaccine, that will be only protective. And that is some people are saying they are still protective with some mutation, but they never said these mutation and what is their effect on the by immunogenic assay. I will show you some essay, but so they hiding the fact, and actually possibly UK started with mutant virus vaccine preparation. I hope USA also started. If they not, immediately start vaccine development with the mutant isolate as I show you. And here I looked at some of the mutations. My point is, if the receptor binding site which bind to the EC2, there is mutation, then I show you some. There is some mutation is going on. It is possible that I show you some of these in other variants. I got same result. Another analysis. We see same result. So and also that dominant mutation P323L RNA dependent RNA polymerase mutation. You see, all are L. They mutated and they are the death signal. So another death signal, I told you that I told the NSP2 is the RNA topoisomerase, and that is very much targeted. We see that people told that T821I, here I show you the some clustering here, T and I mutation, they are death signal. If that mutation is there, that indicated that patient will be died. So that is another mutation, and also that mutation very much in Australia in another mutation in the 120 position 8 phenylalanine into isoleucine into phenylalanine. That paper also published. And that not only this, we see some clustering in the NSP3 protease. The clade G8 is prominent in the USA with the D640G and WRFA Q578. So this WRFA Q578 is the dominant mutation, and there is a death signal. So not only the S protein, we are showing some of the data, the other protein also even in the process. Also, we show you recently, we see LSD deletion in the LSP6 protein of coronavirus, but I don't know if that has any association with the death signal or virus infectivity, but we can check. Now, the Indian and clone. So I must show you here some of the clone here. P484Q in USA, that is K. That is K. But in AL450R, it is the only Indian clone, but now it is found in UK. And this mutation is there, and but they never they told D648Q. Look, they here only show one mutation. They are now in the USA so much mutation, and here also some mutation is there, but it also in the other part of the country different mutation. They showed that this mutation how immunogenic peptide means drugs immunoepitope monoclonal drug, they have different binding, but I cannot show you how much they different. They through the computer analysis they can show some different with the three immunogenic peptide binding to different mutant of the Indian origin virus, double mutant virus. So I just only show you, and here L452R in the domain of the receptor binding domain that also found in the USA. And these recently, as I got some data, and are sequencing. You got some hit here so that you can understand they are there and other mutant how they are changing from one to about 200 mutations. Then they got one very notorious mutation. It is not so easy that they got the notorious mutation so much easily, unless they in March they got the D640G mutation, and also at the same time the P323L mutation in the RNA dependent RNA polymerase. So you show here we show that how those mutants can move to USA, but I don't know much about those. This is some of the paper I mentioning you. Some from Australia, from the pangolin to human to they move here and then here. Whatever they study, there is a lot of sequencing there. But clearly they mentioned there is a peak and they are now increasing in the population and they are immunoescape mutant, which I show you here that L452R virus replication, so virus titer is much higher. And they do the all sequencing and you see the replication also increase. And one question here, the comorbidity. And you see here, all the data they show that there is some comorbidity like diabetes, hypertension, cardiac disease, cancer also there. So it is very evident that if you have comorbidity, then your infection will be increased and death will be maximum. That's also there. So so you and in India, there is a report that they have got much more, 1,494, and they are not, but they are not deposited in the NCBI. That's why I won't get. I have no access to GIS and then United Kingdom also there is not. You see, there is also USA, but I got only one mutant, double mutant. I have not seen in the USA. My data is very few, so I don't know how they got all those things. But you see here, arrival in the India October 5, 2020. Then in the United States February 22 and February 23 in the USA. Then February 26 in the Australia. Then March. Then April. So you understand why there is much here and less there. So if they reach, that means they will do devastating, and India is going devastating now. I hope US government should help India by giving some vaccine with mutant and SD. And that way, I hope India has just started vaccination and there is no vaccine in the hospital because of the preparation is less and sometimes purchase is less, sometimes monetary problem in Indian government. I don't know whatever that is, but I fear. When I saw the TV, my tears come from eyes because what is going on? And it is the 130 crore people. So there would be huge loss. And recently, Lancet, our best journal in medicine, and Nature, the overall best journal in the world, they told that our government not doing a mess, and they should take care the people. So that I just give you some hints of the what is going on. Now, some Indian papers they show that you see that in India there is much less sequencing. There you see the North America, South America, Europe, huge sequencing in the Europe. But in India, there is much less. And in Kolkata, only 54. It is astonishingly low. And sometimes that is partial sequencing. In India, I don't know how much full length sequencing in India. So that must be also who should insist India so that they must do much sequencing and authentic sequencing. And you see the different cloud value G, GH, GR. G1 means G mutation, 640 mutation on the twice it. All here, all they are much increased. So, so here we see there is the clade mutation is different. There is increasing trend. You see red increasing, increasing. Those are the GR. Means N mutation G 240 N40 nucleotide plastic protein. There is increasing here. Open reading frame 3AQ 57H2 dominant. These are one of the flawed nomenclature. Now, whatever the saying, our answer is mutation is selective, and they are claiming life. So we need. This is the paper. This is not actually paper. This is the preprint Biorex TV Indian paper. Another Indian paper. They also this nomenclature is no longer good now. All plot value will be changed and new. Now here I give you some of those plot value. Those are predominant. I'm giving some old value, and here look, I want another mutation. This is the nucleocapsid protein. Here is coming here, another here. So mutation, mutation, and mutation. And here is the N protein, another cluster I show you. That is D, N protein D3L mutation. Surprising, and I show you some of my publications that is not important publication, but I did to show you some of the function of the non-structural protein, not the spike protein. But I am showing you some of the spike protein mutation because which is mostly important. And India, although developed the Covaxin whole attenuated coronavirus vaccine, but we have not got it. I don't know where it is going. Maybe going into USA or UK or in Brazil, but we have not seen it very much. And possibly this time will be India will give much more Covaxin. But I also taken the Covaxin, Covishield, and most of the people taking Covishield in Kolkata. So I know this vaccine has problem because the mutant virus is appear. But here India N501 mutation associated with those double mutation and has not been talked about. If these two mutation and N501 Y mutation and the 614 G mutation must be there. So if these four mutation and I show you other mutation are coming in the but they have not selected much. They have the minimum impact, but who knows? They are the notorious one. So, so U.S. government must do the new vaccine and inoculate it India, because I hope the research in India is far behind and will stay far behind because of the problem of the government, monetary problem, and other problem. I don't know what, but we see have not seen much scientific activity in India. Only they follow USA and UK, and maybe they will follow now China because they are now becoming superpower. But original research in India is very much lacking. That's why we are losing 4,000 people every day. This is same to Indian government and Indian people, Indian scientists, and simple. Even wood is not available. You know, for the burning those dead bodies, they throwing in the Ganga River, and we all drink Ganga River. So this is the very much sad talk and sad to say that our government failed. But I hope US government should help us highly. Otherwise, as according to nature and Lancet, we will lose another 8 lakh people between this 1 month. Within this 1 month, we will lose another 4 times people just due to drugs, oxygen, vaccine, and this is very sad, very sad. Thank you.
Thank you, Dr. Asid. It was a good presentation. Is anyone having any questions? Okay, thank you once again. So we are going to the next presentation. It is from Dr. Asma Haq, Government College University, Faisalabad, Pakistan. Hello, Dr. Asma.
Hello.
Yeah, are you ready with your presentation?
Yeah, I'm ready for my presentation.
Yeah, I'm going to make you the presenter. So first you need to click on the screen button on the below. Yeah, you have 4 buttons on the below of your screen. Click on the 3rd button. Your voice is not audible, Dr. Asma.
Yeah, it is now
Yeah.
unmute. But I'm not allowed to share the screen. I'm constantly pressing the button.
Okay, I have making you have the presenter now. Is it asking for any permissions?
Yeah, it is nothing. Okay, applications, use confidential. Okay, just a minute.
Okay.
Maybe I have to window.
Yeah, it's visible. Yeah, you can start your presentation.
All right. So screen sharing is now paused. So why it is being paused now? Just a minute. Is my screen with the presentation visible?
Yeah, it is visible.
Okay. So, but I don't know what's going on here. Just a minute. Okay. This. Okay, we will talking. We will be
Seeing any
talking.
problem?
Yeah, there is some problem. I'm not sure. There is the indication is that screen sharing is now paused.
No,
So,
we can see your screen, but there is no presentation. It is like a folder wise.
there is no presentation. I know. Just a minute. I think I'm rejoining the meeting. Just a minute, please.
Yeah, okay, no problem.
Yeah, is it
Yeah,
screen
yeah,
visible?
yeah. Yeah, doctor, it's visible.
Okay, fine. Okay, we are actually talking about the, this is basically, you can say, somewhat informative seminar. I have compiled some data regarding this one. So we will be talking about the pivotal role of vaccines while stepping into the post-antibiotic era, right? So first of all, we have to look about what is the antibiotic drug resistance. So actually, this is the ability of bacteria, as most of us know this, that is to resist the effects of the drugs, that these are not killed, right, or their growth is not stopped. So the antibiotic resistance is occurring actually everywhere in the world this time. It actually compromises the treatment of infectious diseases, as all of us know that we have not getting the drugs, the advanced drugs, then first these are first generation and second generation, third generation, and so on, and that these undermine many other advances in health and medicine. So, what is actually an antimicrobial? This is a broader term, and antimicrobial is a substance that kills or stops the growth of the microorganisms such as bacteria, fungi, or protozoa. Or antimicrobial drugs, these include if they kill microbes, we call them microbial, and if they prevent at least prevent the growth of the microorganisms, then we call them the microbiostatic. So scientifically, antibiotics are only those substances that are produced by one organism that kill or prevent the growth of another microorganism. But in common usage, the term is used to refer to almost any drug that attempts, to rid our body, that is a bacterial infection. Most of the time, we see it in the perspective, in the context of the bacterial infection. So this is a compilation of the antivirals we have antibacterials, we have antifungals, we have antiprotozoal, even antihelminthic and anti-algal antibiotics. So what we need to know actually, that's the WHO's first global report on antimicrobial resistance, with a focus on antibiotics resistance, which revealed that it is no longer a prediction. What is no longer a prediction is that the antibiotic resistance, a complete antibiotic resistance, when bacteria will change and ultimately the antibiotics will fail. And this is happening right now across the world. And what does this mean? This is very dangerous. Without urgent action, we are heading for a post-antibiotic era in which common infections and minor injuries can once again easily kill. If you don't have a remedy, then obviously the minor injuries and infections will be sufficient to kill somebody. Then, what is an antimicrobial drug resistance? Antimicrobial resistance is the ability of the microorganisms to withstand the dose of the antimicrobial that was one in the past, that was sufficient in the past, and but due to the repeated exposure of a microbe, a particular drug becomes fails. So reservoirs of resistance may also be present in healthy persons and humans in the animal populations, but they can serve as the carrier. So as we are talking about the antibiotic resistance, this is the background of this talk. Antibiotic resistance is occurring everywhere in the world, and it compromises the treatment of infectious diseases and undermining many other advances in health and medicine. So it occurs naturally, but what's happening right now is due to the misuse of antibiotics in humans and animals, it is continuously accelerating the process. In general, a trend is being observed with time. It takes bacteria to develop antimicrobial resistance, that's AMR, after production of a new antibiotics. It is getting shorter and shorter with the time. So this is the bacteria. Bacteria are very fast in countering, in finding a remedy to the new drug you have discovered, you have synthesized. So they will inevitably find ways of resistance, right? What is drug resistance? Two ways to keep new resistance from developing and to prevent the resistance that already exists from spreading. So this is our purpose, but with every passing day, bacterial infections are becoming more difficult to treat. So, and now we see that we have multiple drug resistance. It is very easily found everywhere. What is multiple drug resistance? It is the resistance of bacteria or say one species of bacteria to more than 2 or 3 drugs at the same time. It means that these drugs are failed. So studies have revealed that the choice of available antibiotics has become very limited because very fast evolution rate in microbes, as we discussed, and the careless use of drugs. So what are the mechanisms of resistance developed by bacteria? So I think this is, yeah. So I will come to the XDR. Now after MDR, that is the multiple drug resistance. I call that the drug resistance to more than 2 or 3 drugs. So now we are commonly hearing another term that is known as XDR, extensively drug resistant. That the bacteria that are classified as XDR, these are almost these are resistant to almost all the present drugs. So this is the extremely dangerous situation. Okay? Likely in the case of the Mycobacterium tuberculosis. So we will have to obviously see what could be the actually how can we manage on the individual level and the government levels. So the post-antibiotic era is actually in fact, it is the end to the modern medicines as we know it. Like I have told earlier that if no antibiotic, if no remedy is available even to a very minor infection, so it can aggravate and eventually it can lead to death as well. So it is just we can assume that what may happen in the 2050. So what were the problems mainly in the 2019? That was the cancer was the major reason of deaths, then diabetes, as you know, then diverse diseases, and then it actually varies from region to region in the world, but this is cumulative. Then the antimicrobial resistance was till 1 or 2 years back, it was like 0.7 million deaths per year. But what you see, what we can anticipate in the 2050, you can have deaths with the AMR, antibiotic resistance. You can have 10 million deaths per year. So this is a very horrible sort of calculation. On the other side, we see that during the last century, life expectancy has increased thanks to the introduction basically of antibiotics. No doubt, the antibiotics have a major role. Hygiene practices and also the vaccination. These contributed to the cure and prevention of many infectious diseases. So, what are the remedies of antibiotic resistance? Vaccines is playing a major role, and there are some other advanced technologies, right? So there are a number of well-established tools to reduce AMR. This again, I will compile it. That is sanitation and hygiene, then funding mechanisms to develop new classes of antibiotics, to win the race against the bacterial evolution. Then antibiotic stewardship, and then education, of course, to avoid inappropriate antibiotic use to treat viral infections. Sometimes we are using antibiotics to treat the viral infections, which is obviously not a wise thing until and unless you have a secondary bacterial infection with that. Then elimination of routine antibiotic use in livestock production. Livestock, when we use antibiotics in the livestock and we consume them, obviously we are the drug resistance through these microorganisms are transferred. So these interventions all have shown benefit whenever and wherever implemented. But it is less well known and much under recognized that the vaccines are also highly effective, valuable tools to fight against drug resistance, or AMR. Vaccines can reduce the emergence and the spread of AMR. It can do directly or even indirectly. So you can see this figure, how much it is in the production of vaccines is important. This is the decline. You can see with the 1910 to 2020, we have seen how much the vaccine production is increased, and comparatively antibiotic production has decreased. So this implies the importance of vaccine production for fighting AMR. So how disease production, prevention by vaccine, how disease prevention by vaccination lowers antibiotic use, and how does it reduces the AMR? So vaccines work by training your immune system to recognize and respond to a pathogen by mounting a rapid and effective immune defense, right? As you know, this is prophylactic mechanism. So this is the way it prevents the establishment of infection or disease, or decreasing disease severity if it has occurred. Okay, so many vaccines have an added benefit and can protect unvaccinated individuals or subjects that cannot be vaccinated in the population by a process that is known as the herd immunity, which reduces the disease in the overall population. Okay. While considering the role of vaccines in fighting the AMR, antimicrobial resistance, and other, this is a strategy other than the antibiotics use. So we have success stories regarding this. So first of all is the disease prevention by vaccination lowers this is very common thing, that if you use the vaccination, you can accelerate the disease prevention. Then there's an example of the Haemophilus influenza type B, HIP. This is a very well-known vaccine, and the pneumococcal, these vaccines are instructive examples in this case. So before introduction of effective conjugate vaccines, so HIP diseases in the children below 5 years of age range from 3.5 to 601 per this number of population in the many countries of the world. But after a study, increase in Hib beta-lactam resistance was observed since the early 1970s, mediated by bacterial expression of beta-lactamases and, or to a lesser extent, modified penicillin binding proteins. There are two mechanisms in case of when aminoglycosides are used. Either the beta-lactam ring is targeted by the bacteria that produce such enzymes which are known as beta-lactamases, and the drug remains then renders ineffective, or other to attack the penicillin binding proteins. There are some proteins, PBPs, the bacteria, which affect the target of the drug. So the one global surveillance study found that 16.6% of all the Hib strains were beta-lactamase positive, with large variation between countries. So this was the data. And But this very horrifying picture changed dramatically after the introduction of, this somehow disturbing. The introduction of the hip conjugate vaccine. So diseases dropped steeply after the introduction of the routine use of hip conjugate vaccines. So you know that immunization programs, this is a must, the hip vaccination for the children. This also led to a significant decrease in the beta-lactamase positive strains. So what you see with this vaccination, that the resistant strains which were actually producing beta-lactamase, so these strains were also drastically reduced. So a similar picture emerged when the introduction of conjugate vaccines to prevent pneumococcal disease. This is also a big success story in this regard. So what happened? In the 1990s, before the introduction of the 7-valent pneumococcal conjugate vaccine, that PCV7, in children, about 63,000 cases of invasive pneumococcal disease occurred each year in the US. So it is known as the PCV7 because this conjugate vaccine has a solution of saccharides which have a different 7 types of antigens. At this time, resistance to penicillin and other classes of antibiotics also spread in streptococcus pneumonia in the US, with invasive pneumococcal becoming resistant to 3 or more drug classes. So this was against the situation that the resistant strains were resistant to 3 or more drug classes. So it's like similar to the hip vaccines, pneumococcal polysaccharide conjugate vaccines proved to be highly effective with more than 90% efficacy against invasive pneumococcal disease. The invasive pneumococcal disease is known as shortly you will call it the IPD. The second generation pneumococcal conjugate vaccines with extended serotype coverage such as like PCV10, this is the next, this is the advanced version. Now with the 10, it is 10 valent, and even the PCV13, the 13 valent, right, have increased direct protection and herd immunity to the pneumococcal serotypes covered by these vaccines with similar efficacy. So then observed after introduction of PCV7 and PCV13 also reduced antibiotic use, and in parallel the prevalence of antibiotic non-susceptible strains also decreased. Not only the antibiotic susceptible, but antibiotic non-susceptible strains were also decreased. So it means that we stated in the initially that directly or indirectly the vaccination reduces the antimicrobial resistance. And it is obviously very important, specifically when you see that antibiotics are getting failed. So it is the rates of antibiotic non-susceptible invasive pneumococcal disease in less than 5 years of age, which is the data collected from 2005 to 2013. You can see here different antibiotics in the different colors, that are the macrolides, that the cephalosporins, there is a tetracyclines and penicillins, and you can see that there's a steep decrease. Right. So these were the vaccines against the bacterial diseases or infections. Now we will see the impact of viral vaccines on AMR. So viral vaccines look something relatively different. So viral vaccines are also very effective in reducing antibacterial resistance. So see, we can view that the diseases or infections are the bacterial, and the resistance has been acquired against these. But which thing is getting effective? That is the viral vaccines. So influenza vaccines do not only prevent influenza infections, but also decrease the likelihood of secondary bacterial infections such as pneumonia and otitis media. And you know, when there is a secondary bacterial infection in the presence of a viral infection, how much grave the situation becomes. So there are two major mechanisms by which even moderately, you can see seasonal influenza vaccine can reduce the antibiotic use. So firstly, they prevent secondary bacterial infections. as I told you, and thus the use of antibiotics to treat these infections, right? Okay. A study conducted in the Canada in the year 2009 demonstrated convincingly that influenza-associated antibiotic prescriptions declined by 64% after introduction of universal influenza vaccination in Ontario compared to other Canadian provinces that limited the use of influenza vaccines to populations most at risk of disease. So this is a really a big contribution to this vaccine. So in another study conducted in Turkey, that is relatively an older study, but they studied that influenza vaccination significantly reduced acute otitis media in children by 50.9% compared to unvaccinated controls. Okay, this was the first. Then the second mechanism by which viral vaccines can reduce AMR is by prevention of inappropriate antibiotic prescriptions for respiratory tract infections caused by viral pathogens. In the US, for example, half of all antibiotic prescriptions are inappropriately written for respiratory illnesses associated with pathogens such as influenza, respiratory syncytial virus, RSV, and other viruses that are not susceptible to antibiotics. But these examples illustrate that even viral vaccines can play an important role in reducing antibiotic use that drives bacterial resistance. We have lots of data regarding this. So if interested, we can find very reliable and recent data that implies the role of, emphasizes the role of the viral vaccines in combating the bacterial infections. Now, we will consider some vaccines that are in late stage development with potential to reduce AMR. Clostridium difficile. It is an anaerobic spore-forming gram-positive bacillus and the main cause of antibiotic-associated nosocomial and community infectious diarrhea. It is a real mess. The incidence and disease severity have increased over the last decade. In the US alone, approximately 500,000 cases of Clostridium difficile infection, we will briefly describe the CDI, are diagnosed annually, along with 29,000 associated deaths. The CDC has classified C. difficile as an urgent threat. This is an urgent threat based on the burden of its infection and the rapid global spread of epidemic and antibiotic-resistant strains. Okay. Once the infection occurs, the patient is often caught in a vicious cycle. Antibiotic use, then apparent cure, but subsequent recurrence of the disease that then again has to be treated with antibiotics. That is like a never-ending cycle of a disease starts in this way. Sometimes when you get treatment, apparently it looks all okay, and you leave the treatments, and then again, okay. The CDI is mediated by toxins, which can be inactivated by toxin neutralizing monoclonal or polyclonal vaccine elicited antibodies. The mechanism of action or pathogenesis of this toxic-based disease is clearly understood, which has proved to be very helpful. And the vaccine-induced generation of toxin-neutralizing antibodies has proven successful for a number of other toxin-based diseases as well, like tetanus, anthrax. So, the generation of the toxin-neutralizing antibodies, whether monoclonal or polyclonal, this mechanism has been proven very helpful, not only in this particular case, but for other toxin-based diseases. So at least two Clostridium difficile vaccines are in phase 3 clinical development, and that looks very promising. Since CDI incidence increases with age, an efficacious vaccine to protect all adults 50 years and older would be highly desirable. So such vaccine would be an additional highly effective tool in reducing antibiotic use and associated AMR, as vaccinated individuals should not contract CDI thus reducing the spread of spores and bacteria, the associated antibiotic use, and the generation of antibacterial species. This was the one case, and second is the Staphylococcus aureus, a very well-known pathogen that is known as a superbug. Actually, I have recently closed a research over it. I have studied its resistance. It's highly resistant in our populations. We have found not only the MRSA, the methicillin-resistant Staphylococcus aureus, but also the VRSA, vancomycin-resistant Staphylococcus aureus. Although in some countries we are only finding the VISA strains of Staphylococcus aureus, we have reported several VRSA isolates. So this is very rightly known as a superbug. Can you listen to me, please?
Now, yes, but before no.
I think there was some connection problem. Should I continue?
I think yes.
Okay. Okay. I was talking about the MRSA, the methicillin-resistant Staphylococcus aureus. It contributes to over 70,000 cases of invasive diseases per year in the US alone. And now this superbug is not only reported in the nosocomial infections, it is much reported in the community settings. So this is the point of concern. So both the CDC and the WHO have recently placed MRSA on their antimicrobial resistance watch lists, highlighting the significance of AMR in this species. S. aureus has the propensity to develop resistance even to newer antibiotics that have been introduced, such as linezolid and daptomycin, and it happens so fast. We have studied this in the linezolid, and as I told you, we have also found the vancomycin-resistant strains, VRSA strains, in our population. As aureus glycopeptide resistance has emerged as a source of concern, as this class of antibiotics, including vancomycin, is one of the main resources for combating infections caused by MRSA. With the often uncontrolled empirical vancomycin use, particularly during elective surgeries, the resistant rates could be further driven, which has already been documented by the emergence of vancomycin resistance. So the point of discussion is that the only remedy among the antibiotics till now is the vancomycin, and we are finding resistance against it. Firstly, we were finding the VISA strains, and now in some countries, VRSA strains are also reported. And when the vancomycin is getting failed, what next? So this is really the point of concern. So So given the disease burden and adaptability of S. aureus to develop resistance to many classes of antibiotics, the development of effective vaccines would be highly desirable in curbing disease as well as reducing AMR. So vaccines that are currently in clinical development differ from the earlier vaccines in that they are designed to address more comprehensively the complex pathophysiology of S. aureus infection by eliciting antibodies that target multiple virulence factors. So forcing the many appearing problems and the quickly acquiring resistance in the S. aureus, the newly vaccines being designed unlike something multi-purpose. So multiple that they can target multiple virulence factors. So the most advanced vaccine candidate that includes a 4-antigen S. aureus vaccine that is known as the SA4 AG for the prevention of invasive S. aureus disease in elective spinal surgery patients. And this vaccine was shown to be safe, well tolerated in clinical trials, and induced high levels of bacterial killing antibodies in healthy adults, leading to a fast track designation by the US FDA. And another recent development is the 4-component S. aureus vaccine, that is 4C step, formulated with the TLR cell-independent adjuvant. An adjuvant has been added to it, which is the TL4 cell-dependent. So this is, you can see the potential correlates of immunity for an effective Staph aureus vaccine. Though these are the development, but this is how can we understand is the ideal S. aureus vaccine would comprise appropriate antigens, okay, yes, antigens and more antigens, more effective, an adjuvant capable of driving adjuvant that can make the function of the vaccine more fast. It gets more effective and speedy in action. An adjuvant capable of driving both antibody and T cell responses. Antibodies are required to neutralize secreted toxins. These are neutralized by different mechanisms. Antibodies neutralize these secreted toxins and facilitate phagocytosis by opsonization of the bacteria. This is another immunological process that outer any foreign substance, the phagocytosis is carried out. So these facilitate antibodies help in these two functions. Then there is the induction of the memory T helper cells Th1 and or T helper cells 17 Th17 cell responses that would in turn recur and activate phagocytes and leading to the destruction and removal of the organism, the pathogen, right? The S. aureus. So induction of follicular T helper cells, TFH, is also desirable to promote antibody responses. Then ABC, antigen presenting cell, right? You can see here in left side in the star-shaped blue body. And the chemokine ligand, then IFN gamma, interferon gamma, iron, interleukins, PRR, pattern recognition receptors, SCBSI, Staphylococcus aureus, bloodstream infection. So these, in a successful vaccine which we wish to design, which is desirable today, we keep in mind that the multiple targets, we want to hit the multiple targets. So then we come to the M. tuberculosis. The hunt of effective vaccines has been challenging. It is very well-known example is the M. tuberculosis. So we know we were very very well aware of the BCG. It's always have been added in the vaccination program. You know, the BCG vaccine that stands for Basil Calmette Curine, after the name of the scientist. In use, it's been used for almost 100 years. It's composed of an attenuated strain of the Mycobacterium bovis and used to vaccinate more than 90% of newborns in endemic countries because it is already since long it has been added to the vaccination programs for the newborns. So attenuated strain is a strain that is live strain, but the whole organism is present, but it has by culturing and subculturing, you have weakened it to such an extent that it is no more dangerous to a normal person, to a healthy person, or less immunocompromised persons. The shortcomings of BCG make the development of more modern vaccine approaches a priority, but because it is a very old remedy and there are many changes and resistance has been increased during that time and other changes have been there. So we are finding many shortcomings in the BCG. So many tuberculosis vaccine approaches have been evaluated under evaluation, and as of today, none have been successful, but there are many under trials. So one promising approach that is still in preclinical stages of evaluation uses a rhesus cytomegalovirus vector expressing multiple TB antigens, right? So this is a vector, cytomegalovirus vector, which is being used in this approach. By this approach, either complete protection against TB challenge and disease in some rhesus macaques or partial protection was observed after immunization. So the data are very exciting as the rhesus TB challenges were conducted with highly virulent TB strains that is designed to also demonstrate long-lasting protection in this model. So the latest approach we followed was started in 2019, but nothing is, but still these all are under trials. If such rhesus, yes, sorry. In such rhesus cytomegalovirus TB-based vaccine constructs were to provide meaningful protection in humans in the future, the impact on AMR would be very significant given the high level of AMR exhibited by this organism and the difficulties treating patients with the disease requiring a cocktail of antibacterials. So you have to do a lot of it in trial with the TB patients still, and still it is a very life-threatening disease, particularly in some certain countries. What will be the future prospects? It is unlikely that there will be vaccines that can protect against all the bacteria that are contributing to the AMR, because these are not particularly designed for this purpose. Of course, these are prophylactic. You are initially somehow controlling the spread of resistance in the bacteria, or you're controlling the resistant strains. So this is naturally not possible that you can design a vaccine for every such threat, bacterial threat. You have to drugs are of course the main counter. So the prospects for vaccines in later stages of development are encouraging to do their part in fighting AMR once shown to be effective and appropriately implemented. But this is very prospective, you cannot deny it. So if done with the appropriate planning, it can be very fruitful. To provide maximum public health benefits, there must be efforts to steadily increase vaccine coverage as well as complete study and licensure proceedings of new vaccines addressing important bacterial and viral pathogens for which vaccines currently do not exist. So maximum public health benefits, you have to adopt all the strategies, and this also includes that efforts should be continued and increased to steadily increase vaccine coverage as well as complete study and licensure and all the stages that are involved in this. Newer vaccines under development, as I have discussed, that the vaccines are to prevent Clostridium difficile and the Otis aureus and the pneumococcal conjugate vaccines, which extend serotype coverage, and the vaccines to prevent infections with gram-negative bacteria. Okay, so vaccines, the gram-negative bacteria, by the way, I will tell you that these are the most of the pathogens, bacterial pathogens belong to the gram-negative group of bacteria, and these are very threatening. And then tuberculosis. Okay, these hold the newer vaccines which are under development. These hold a profound promise to not only address these life-threatening diseases, but to help further antibiotic use and therapy to prevent the AMR. But there is no limit to how many vaccines can be given to an individual, as humans are exposed to a continuous basis to a myriad of infectious diseases agents, and the immune system has been developed to deal with an almost infinite number of pathogens. You can never estimate the number of antigens you are exposed to. So here is no limit, okay? For more antigens, as you know, the pathogens also contain the antigens, and a single pathogen may contain many antigens at the same time. So how can we limit? We cannot estimate the extent how much vaccines can be needed. So this is a continuous, you can say, a journey, and there will be many questions that can arise for time. And of considerable importance will be an emphasis to reduce antibiotic use in animal and fish husbandry to wisely control the, you can say, the antibiotic spread, the AMR spread. You have to reduce at least the one challenge you can manage, that in the animal and fish husbandry, which you can consume as food, and you get the antibiotic resistance directly from them. It can be controlled over here. And the development of veterinary vaccines could help to stem the inappropriate widespread prophylactic use of antibiotics in animals. So if you use wisely in the animals, the veterinary vaccines, if you can manage it, so it can also be very helpful. The importance of maintaining and creating new markets for vaccines cannot be understated, of course, because this is a huge industry. It is vital to provide incentives to vaccine companies to develop vaccines that are of public interest but may not be commercially viable. Sometimes, if it's not commercially viable, it needs support, whether it is the private sector, whether it's the government, it needs huge funding for the vaccinations, and it has to. So what can we do for the approaches? We can quickly look at this, that the continued improvements to sanitation and the hygiene, that the specific disease prevention guidelines from medical societies, as we know, improved funding mechanisms for anti-infective and vaccine research and development. Of course, this is the big thing. The incentives for manufacturers to invest in anti-infective and vaccine research and development, then continued education programs for prescribers on antibiotic stewardship. This is also very important. Then continue initiatives globally to moderate, eliminate, where possible, the routine use of antibiotics in agriculture and aquaculture. So I'm really acknowledged to these scientists for the preparation of this presentation. So, any questions?
Thank you, Dr. Asma. It was a nice presentation. Is anyone having any questions? I think there are not. Thank you once again. So by this, we came to end of our day 2 webinar. I would like to thank all the attendees and participants of today's webinar. Hope you enjoyed the webinar and gained knowledge from the sessions. If you have any queries, you can mail us to the concerned program manager, and we'll be glad if you could join us in our next webinar, which is scheduled, and the dates will be informed to you by mail. We'll be happy if you could share your opinion or feedback with us. Thank you once again, one and all present here. Have a nice day.